are related. Now next up, we have enzyme terminology. And before we talk about a bunch of specific enzymes, we'll need to learn a little terminology. Now the name and the function of an enzyme are related, and if you understand this nomenclature, you can figure out the basic function of an enzyme. Now we're going to briefly cover some important enzyme categories. We'll start off with our first category here, the kinases. Now these enzymes transfer a high energy phosphate group from ATP to a target substrate. An example of a kinase is phosphofructokinase, as you can see here. Now you can find phosphofructokinase in the glycolysis pathway, where glucose is broken down to create ATP. And here, phosphofructokinase phosphorylates or adds another phosphate to fructose 6-phosphate in glycolysis. And this will form fructose 1,6-bisphosphate, as you can see here. And just to recap that, fructose 6-phosphate is acted on by phosphofructokinase, which takes a phosphate from ATP and sticks it on fructose 6-phosphate to form fructose 1,6-bisphosphate. The thing to remember here is that kinases transfer a high-energy phosphate group from ATP to a target substrate. Now next up, we have the phosphorylases. And what do you think they do? Well, the phosphorylase enzymes are similar to kinases, meaning that they add a high-energy phosphate group to a substrate. The difference here is that this enzyme does it without using ATP. An example of a phosphorylase would be glycogen phosphorylase, which is found in the glycogenolysis pathway. Now, glycogenolysis is where you take a large glycogen chain and break off free glucose molecules. And here, glycogen phosphorylase grabs a free phosphate and cleaves the 1,4-glycosidic bond in glycogen to produce glucose 1-phosphate. So what do you need to remember here about the phosphorylases? There you go. They add a high-energy phosphate group without the use of ATP. Next up, we have the phosphatase enzymes. And these are the opposite of kinase and phosphorylase enzymes. Phosphatases, like fructose 1,6-bisphosphatase, remove a phosphate group from a substrate. And here, fructose 1,6-bisphosphatase converts fructose 1,6-bisphosphate to fructose 6-phosphate. And this reaction can be found in gluconeogenesis. And in this process, we saw a substrate with 2 phosphates attached. And after enzymatic removal, the product only has 1 phosphate. So once again, the important point here is that phosphatases cleave off phosphate groups. Now next up, we have the dehydrogenases. And these catalyze oxidation-reduction reactions. And while we're on it, do you happen to remember what a redox reaction is? There you go, it's the exact same thing. It's an oxidation-reduction reaction. And this means that hydrogen and its corresponding electrons are removed from one substrate and added to another. An important example of this is pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA, as you can see here. Also here, you'll see that pyruvate dehydrogenase is moving electrons and protons to form NADH, which will then be used later. The big thing that you need to remember here is that dehydrogenases catalyze oxidation-reduction reactions. And this just means that hydrogen and its corresponding electrons are removed from one substrate and added to another. Now next up, we have the carboxylase enzymes. And these move carbon dioxide groups onto a substrate, as you can see here. A great example of a carboxylase enzyme is pyruvate carboxylase. And here, a pyruvate molecule has a CO2 group added with the help of pyruvate carboxylase. And this forms oxaloacetate, which is a molecule with 2 CO2 groups. Now the carboxylase enzymes need a little help. Therefore, they have a cofactor. And do you know which one this is? There you go, it's biotin, or vitamin B7. So once again, the important point to remember here is that carboxylase enzymes move carbon dioxide groups onto a substrate, and they often need a cofactor, which is biotin, or vitamin B7. Now what do you think you'd call an enzyme that actually removes CO2? There you go, that'd be a decarboxylase enzyme. And as we just stated, the decarboxylase enzymes remove CO2 groups. Now what about the hydroxylases? Well, these are enzymes which end up adding hydroxyl groups, as you can see here. Now how about the mutases? Well, the mutases are enzymes that move functional groups around a molecule. An example of a mutase would be methylmalonyl-CoA mutase. And just to review these one more time, decarboxylase enzymes remove CO2, and hydroxylase enzymes add a hydroxyl group. And mutase enzymes move a functional group from one spot to another on the same molecule. Now next up, we have the synthetases as well as the synthase enzymes. And these work to join molecules together. The only difference here is that one uses ATP while the other does not. Now can you guess which one actually does use ATP? There you go, that's going to be the synthetases, and they use ATP or GTP, while the synthases do not. Now an example of a synthetase is carbonyl phosphate synthetase, an enzyme which converts ammonia, NH3, and CO2 with the help of 2 ATP to carbonyl phosphate, as you can see here. Now an example of a synthase enzyme is citrate synthase, which converts oxaloacetate plus acetyl-CoA to citrate, as you can see here. So once again, the big point to remember for synthetases and synthases are that they're enzymes that join molecules together, and that the synthetases use ATP or GTP, while the synthases do not. Alright, well that'll wrap up enzyme terminology. See you in the next video.
Hi everyone, this is our lecture on the rate-determining enzymes of metabolic processes. So let's get started. To get a clinical perspective, let's look at a quick flash case. Let's say a 22-year-old medical student presents to his primary care physician with fatigue and jaundice. He was last seen in the office only 3 days ago, in preparation for his upcoming volunteer trip to Africa. Let's keep this in mind as we go through the lecture. Our learning objectives are to list the rate-determining enzymes of metabolic processes and to recognize the regulators of rate-determining enzymes and their metabolic processes. Here, we have a table of the different metabolic processes in the first column, the rate-determining enzymes in the second column, and the regulators in the third column. Now, yes, I recognize that this is very overwhelming, but it condenses some of the most high-yield information regarding the processes into one table. The enzymes in particular are some of the most commonly tested enzymes, since they determine the rate at which a reaction can occur. The rate-determining enzymes are also the ones most commonly targeted by drugs, because slowing them down or speeding them up will influence the process they're involved in, which might not be the case for the other enzymes in the pathway. It'll definitely help you to know which enzyme is rate determining for each process, and how these enzymes are regulated. Now, we won't discuss all of these as that wouldn't be worth your time, but we will discuss a few as well as some clinical tie-ins. Let's take a closer look at glycogenesis, or the creation, storage of glycogen, and glycogenolysis, or the breakdown of glycogen into glucose. As you can see, the inducers of glycogenesis are the inhibitors of glycogenolysis, while the inhibitors of glycogenesis are the inducers of glycogenolysis. And this all makes sense looking at the diagram. These two processes are opposite sides of the same coin and are involved in glycogen metabolism. If you have plenty of substrate around in the form of glucose 6-phosphate, you will induce glycogen synthesis. And the same goes if you have insulin or cortisol present. Now, this process is inhibited if epinephrine or glucagon are present, and this is done through protein kinase A, as you see here. Now, one quick tie-in here is that in stressful situations, you want plenty of glucose available for the body to use. This is why epinephrine leads to the inhibition of glycogen synthesis and stimulates glycogenolysis. Another tie-in is related to insulin-dependent diabetes. Here you are providing insulin to help improve blood glucose levels. This insulin is going to stimulate glycogen synthesis and inhibit glycogen breakdown via protein phosphatase, as you see here. Now, patients using insulin are at risk of developing hypoglycemia, and every patient that is given an insulin prescription should also be given a prescription for a glucagon kit. Can you tell me why? Great. that would treat hypoglycemia. Glucagon would stimulate the breakdown of glycogen into glucose, increasing blood glucose levels. Often, in severe hypoglycemia, patients are too obtunded to realize they need to eat, or for a family member to be able to safely give them food to increase their glucose levels. In this situation, glucagon is administered intramuscularly to help relieve the hypoglycemia. Another common example is HMG-CoA reductase, which is the rate-limiting step in cholesterol synthesis. HMG-CoA reductase converts HMG-CoA to mevalonate, which eventually gets turned into cholesterol. Do you remember which class of drugs inhibits this enzyme? Great! The HMG-CoA reductase inhibitors, or statins, which are used to treat hypercholesterolemia. Therefore, statins inhibit the rate-limiting step for cholesterol synthesis. Let's talk about one more, the hexose monophosphate or HMP shunt, also called the pentose phosphate pathway, which parallels glycolysis to create NADPH and products for nucleotide, nucleic acid, and amino acid synthesis. Its rate-limiting enzyme, as we can see in this diagram as well, is glucose 6-phosphate dehydrogenase, often abbreviated as G6PD. Why would it matter if we had a deficiency in G6PD, which is a common human enzyme deficiency and it's X-linked recessive? Well, G6PD forms NADPH from glucose 6-phosphate, and NADPH is necessary to keep glutathione reduced because it detoxifies free radicals and peroxides. Decreased NADPH in our red blood cells would lead to a hemolytic anemia, because then we'd have weak defenses against oxidizing agents, including sulfonamides, anti-tuberculosis drugs, and antimalarials. Infection can also precipitate hemolysis. When the globin chains get denatured and precipitate within the red blood cells, they form Heinz bodies, as you can see here. Great. Let's do a quick flash quiz. What is the rate-determining enzyme of glycogenolysis? Yes, glycogen phosphorylase is the rate-determining enzyme of glycogenolysis. Extra credit if you thought of the regulators too. Well, let's go back to our flash case. Hey, didn't we just see that image? You're right if you're saying those are Heinz bodies from the denatured globin chains that are precipitated in red blood cells due to oxidative stress. But what is stressing out a 22-year-old medical student? Well, besides medical school. He's planning a volunteer trip to Africa, and a few days ago, he may have been prescribed an antimalarial, like chloroquine. If he is deficient in glucose 6-phosphate dehydrogenase, then he doesn't have enough NADPH to defend against oxidizing agents. Well, we've made it to the end. If you're looking for more, do not despair. All of these enzymes will be covered throughout the biochem metabolism section, but this is a great review table, so please make sure you have a firm grasp of these enzymes and regulators prior to test day. What's the bottom line? Rate-determining enzymes determine the rate at which metabolic processes occur and are controlled by numerous regulators. And rate-determining enzymes are frequently targeted by drugs to speed up or slow down a metabolic process. That's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.
Alright, so next up, let's talk about some sites of metabolism. It's useful to group metabolic reactions by the site in which they occur. Most reactions occur either in the mitochondria of the cell or within the cytoplasm, although some reactions occur in both. Now don't be surprised if a USMLE question asks you where a certain reaction takes place. And yes, this is very high yield. Now in the mitochondria, reactions such as fatty acid oxidation, which is also known as beta oxidation, acetyl-CoA production, the TCA cycle, oxidative phosphorylation, and ketogenesis take place. And the cytoplasm plays host to reactions such as glycolysis, fatty acid synthesis, the HMP shunt, protein synthesis, and the rough endoplasmic reticulum, steroid synthesis, and the smooth endoplasmic reticulum, nucleotide synthesis, as well as cholesterol synthesis. Now, as we just stated, protein synthesis takes place on the rough endoplasmic reticulum. And do you remember which type of proteins are produced by the rough endoplasmic reticulum? There you go. These are proteins that are meant to be secreted from the cell, or bound within organelles, or embedded in membranes, as these are all synthesized by the rough endoplasmic reticulum, while the cytosolic proteins are synthesized from free ribosomes. And just to review, where does steroid and cholesterol synthesis take place again? There you go. That occurs on the smooth endoplasmic reticulum. Now, can you name a few reactions that take place in both the mitochondria as well as the cytoplasm? There you go. These include heme synthesis, the urea cycle, and gluconeogenesis. And an easy way to remember these is it takes two to hug. With all that said, if you look closely at these individual pathways, you'll see that part of the pathway occurs in one location, while the rest of the pathway occurs in a separate location. And just to review, do you remember which vitamin is required for heme synthesis? There you go. That'd be vitamin B6, or pyridoxine, and it's used to convert succinyl-CoA and glycine to aminolavulinic acid, which is the first step in heme synthesis. Alright, now let's take a shot at a flash quiz. Glycolysis occurs in the, there you go, cytoplasm, whereas the TCA cycle occurs in the, great, the mitochondria. Now it's important to remember where these two pathways occur. And just to review, glycolysis again occurs in the cytoplasm, whereas the TCA cycle occurs within the mitochondria. Alright, well that'll wrap up CITES metabolism. See you in the next video. Now next up, we have the Summary of Pathways diagram. And if you're like me, this may seem a little intimidating. But it's meant to show you how all the metabolic pathways that we're about to go over are interconnected. Now, we simplified it here on this slide to make it a little easier to interpret. The take-home message is that carbohydrate metabolism is connected to protein metabolism, lipid metabolism, and even nucleic acid synthesis. In this figure, just recognize how everything is interrelated, and we'll talk about each of these processes throughout the lectures as we go. Now, once you feel more comfortable with the various smaller pathways that we'll discuss throughout the section, it would be helpful to look back and see how all of it fits together. Here, we'll slowly introduce enzymes, substrates, and products that will fill in this diagram to make a more complete picture. This is also a great page to take notes from other parts of this chapter or even from other chapters, since there are a lot of inputs and outputs that are actually not shown. An example here is 1,3-bisphosphoglycerate is used to make 2,3-bisphosphoglycerate, which is used in erythrocytes to reduce the affinity of hemoglobin to oxygen, and this will be discussed more in the respiratory chapter. Also, fatty acids are broken down into acetyl-CoA, which can be transported using the carnitine shuttle into mitochondria, where it's converted into acetyl-CoA, which can then enter the TCA cycle. As you study, you'll probably find a lot more things that you can add to this page. For now, don't feel like you need to memorize all of this before you move on. Just realize that this diagram is here to provide a summary of how the metabolic pathways are interconnected. Alright, well that'll wrap up Summary of Pathways. See you in the next video. Alright, so next up we have the activated carriers. And here you can see the table from the book. So what do we mean when we say something is an activated carrier? Well, these are carrier molecules which actually carry something called a functional group. Now, the functional groups here are different depending on which carrier molecule they're associated with. And these functional groups are eventually transferred from one molecule to another during metabolic reactions. The big thing to remember here is which functional group each activated carrier is associated with. This can help you determine the outputs of each metabolic reaction. However, knowing the exact structure is not all that important. Now, the first activated carrier we're going to talk about is adenosine triphosphate, which carries 3 phosphoryl groups. So ATP here is the activated carrier, which is carrying the functional group of a phosphate. And it can use these high-energy phosphate bonds to phosphorylate proteins, which can activate or inhibit the function of the protein, or change how it interacts with other molecules. So the big thing to remember here is that ATP is the activated carrier, which carries the functional group of a phosphate. Now next up, we have NADH, as well as NADPH, as well as FADH2. Now these carrier molecules actually carry electrons. And these electrons can be traded in later down the line to help make ATP. And can you think of an example of this? There you go, in the electron transport chain. Now next up, we have coenzyme A, which you can see here, as well as lipomide, which is a lipoic acid conjugated to a protein via amide bond. This group then acts as a carrier for an acyl group, which you can see here. Now, did that sound kind of dense? Well, don't freak out. All you really need to know here for your test is 2 things. That CoA and lipomide carry acyl groups. And what exactly is an acyl group? There you go. It's a generic name that we give to an RCO group. And these can be found on esters, ketones, and amines, to name a few. And an important example here is acetyl-CoA, which is formed by pyruvate. The acetyl-CoA here acts as an acyl carrier, which can enter the citric acid cycle for ATP production, or go on to be involved in fatty acid metabolism. And just to review, coenzyme A as well as liponamide are carrier molecules which carry acyl functional groups. Now next up, we have biotin, or vitamin B7, which carries carboxyl groups and acts as a cofactor for carboxylation enzymes. And next up, we have tetrahydrofolate. And do you remember what it's formed from again? There you go, folate, or vitamin B9. And tetrahydrofolate here is a carrier of a single carbon group, and therefore acts as a cofactor for single carbon transfer methylation reactions. Another important point to remember here is that tetrahydrofolate is important in the synthesis of nitrogenous bases, which can be seen in DNA and RNA. Now next up, we have S-adenosyl L-methionine, which is also known as SAM. Now this molecule carries a methyl group and is important in DNA synthesis as well as the conversion of norepinephrine to epinephrine. So last of all here, we have thiamine pyrophosphate. And this is the active form of thiamine, or vitamin B1. Now here, thiamine pyrophosphate acts as a cofactor for many of the dehydrogenase enzyme reactions. And it does this by carrying the aldehyde functional group. Well, alright, that's going to wrap up the activated carriers. See you in the next video. These include the nicotinamides, which include NAD+, as well as NADP+, as well as the nucleotides, which include FAD-. Now thinking back to the biochemistry nutrition section, do you remember which vitamin FAD is derived from? There you go, FAD is derived from riboflavin, or vitamin B2. Now how about NAD+, as well as NADP+? Do you remember which vitamin these nicotinamides are derived from? Great! NAD+, as well as NADP+, are derived from niacin, or vitamin B3. Now all 3 of these will accept electrons using various redox reactions, and then they will transfer those electrons to other molecules, such as proteins within the electron transport chain. Now NAD+ is mostly used for catabolic processes that break down organic molecules. and the electrons are carried in reducing equivalents as NADH, as you can see here. Now, if NAD+ is primarily used in catabolic processes, what is NADPH used for? There you go. NADPH is commonly used in anabolic processes, and these include things like steroid and fatty acid synthesis, where it supplies the reducing equivalents, or electrons, and NADP+ is the result, which can accept more electrons. Now, NADPH can also be created using the hexose monophosphate shunt, or the HMP shunt. And NADPH here can be used in several different places, and these include anabolic processes, as we just mentioned, also the respiratory burst to create oxygen radicals to damage and kill bacteria, as well as the cytochrome P450 system, and by glutathione reductase to reduce glutathione. Alright, well that'll wrap up the universal electron acceptors. See you in the next video.
Hey everyone, my name's Christina and this video is on hexokinase and glucokinase. So imagine you're on your family medicine rotation. You finished rounding and charting for the day and now you're sitting in the residency didactics. Today they're going over diabetes and specifically insulin-dependent diabetes. Midway through the lecture, one resident notices how sleepy the medical students look and decides to ask, what enzyme does insulin act on? All the other medical students look dazed, trying to recall M2 Pharm, but not you. You recall clearly, and you raise your hand to be called on. In this video, we're going to be talking all about hexokinase and glucokinase. In the next few slides, you should be able to 1. Recognize the importance of a hexokinase and glucokinase in the metabolism of glucose. 2. Describe the location, affinity, and speed of each enzyme. And 3. Associate relevant clinical disease with hexokinase or glucokinase mutations. Alright, let's dive right into the video. Do you remember what the function of a kinase enzyme is? A kinase enzyme transfers a high-energy phosphate group from ATP to a substrate. So we have a cell here and a molecule of glucose located outside the cell. It is free to enter and leave the cell in its current form. But once a glucose enters a cell, it meets with the enzymes hexokinase or glucokinase, which adds a phosphate group to the glucose molecule. Thus, the first step of glycolysis is phosphorylation of glucose to create glucose 6-phosphate. This step requires 1 ATP. Why is a phosphate added to the glucose? Well, in addition to preparing glucose for the next step in glycolysis, this also gives glucose a negative charge, which now prevents it from passing through the plasma membrane, which effectively traps the glucose within the cell. As stated earlier, this reaction can be catalyzed by either hexokinase or glucokinase. The difference between these two enzymes have to do with their locations and enzyme kinetics. You will learn more about enzyme kinetics in the pharmacology section, especially about the Km or affinity and the Vmax or speed, which you see here in this graph. For now, I'll briefly go over the kinetics of these two specific enzymes, hexokinase and glucokinase. First, we'll focus on hexokinase. It's found in almost every cell type in the body, except for liver cells and pancreatic beta cells. Hexokinase has a high affinity for glucose, but has a low maximum reaction rate. This means that the concentration of glucose needed for this enzyme can be used is relatively low since it has a high affinity, but the speed at which a reaction occurs is pretty low due to its low Vmax. Hexokinase is not affected by insulin, but does receive negative feedback from high amounts of glucose 6-phosphate. So basically, when the cell has low glucose 6-phosphate levels, hexokinase will catalyze any glucose available in order to form glucose 6-phosphate and store it within the tissue. However, if there's a large amount of glucose in the blood, such as after a meal, hexokinase won't be able to keep up since it has a low capacity or low Vmax. Okay, so on to glucokinase. Glucokinase comes into play after meals when there are high glucose loads. Where is glucokinase found in the body? Glucokinase is only found in the liver and in beta cells of the pancreas. This enzyme has a low affinity for glucose, but a high capacity or reaction rate. This means that glucokinase is not going to be active unless you have a lot of glucose, but when you do have a lot of glucose, it can act very quickly to phosphorylate it. Also, it has no feedback inhibition from glucose 6-phosphate, so as long as there's lots of glucose in the bloodstream, glucokinase will keep phosphorylating glucose inside of cells. It does, however, receive feedback inhibition from fructose 6-phosphate. Glucokinase is also directly influenced by insulin. You can think of this as being kind of a buffer system because after you eat a large meal, glucose will activate the beta cells in the pancreas, which will then release insulin, further activating glucokinase in the liver so that the glucose is removed from the blood and stored as glycogen. Another way to think of these two enzymes is like the tortoise and the hare. Hexokinase is the tortoise because it acts at a slow and steady pace, while glucokinase is the hare, speeding up and slowing down depending on the amount of glucose in the blood. I want to touch on the concept of diabetes, and specifically maturity onset diabetes of the young, or MODY. Again, you'll learn more about the disease diabetes as a whole in the endocrine section, but in a nutshell, diabetes results in an increase in glucose in the bloodstream, and there are many different forms of diabetes, type 1 versus type 2, and then MODY. Maturity onset diabetes of the young is a group of monogenic diseases, meaning there's usually only one mutation in one of the few genes causing the diabetes, versus the other forms of diabetes which usually have multiple genetic or non-genetic causes. One of the mutations in MODY can involve the glucokinase gene. This mutation will functionally alter the glucokinase enzyme, producing a form of diabetes. Now, why is that? Remember, glucokinase is induced by insulin when there are high levels of glucose in the bloodstream. If a point mutation causes the glucokinase enzyme to malfunction, insulin will no longer activate glucokinase like it normally would, resulting in increased levels of glucose in the bloodstream because glucose would no longer be trapped inside the cell. It's important to note that hexokinase has not been associated with MODY. Can you think of any diseases that involve hexokinase? One such disease is essential fructosuria. Now, we just went over how hexokinase and glucokinase play a role in glycolysis through phosphorylation of glucose 6-phosphate, but this specific disease involves the metabolism of fructose. It is due to a defect in the enzyme fructokinase, not hexokinase itself. Fructose is typically first converted to fructose 1-phosphate via fructokinase, and will eventually enter glycolysis. But in this disease, fructokinase is defective. So if fructokinase is not functioning, then fructose is metabolized by hexokinase, which converts fructose to fructose 6-phosphate, which can then proceed through glycolysis. This is a rather benign disease, and clinically, you really only see fructose in the blood and urine. Don't forget to check out our video on fructose metabolism. Okay, let's do a quick flash quiz. Piggybacking from our introduction scenario, a 65-year-old with type 2 diabetes injects insulin before his lunch. How does insulin affect the action of hexokinase and glucokinase? Hopefully you remember that insulin induces the enzyme glucokinase, but doesn't affect hexokinase. This is because hexokinase is always active, while glucokinase must be stimulated by insulin when glucose levels are high in order to create more glucose 6-phosphate to trap glucose inside the cell. Okay, so the bottom line is, hexokinase and glucokinase are involved in glucose metabolism by phosphorylation of glucose to create glucose 6-phosphate. Hexokinase functions in almost all tissue, except the liver and pancreas, while glucokinase solely functions in only the liver and pancreas. MODY, or mature onset diabetes of the young, is a subset of diabetes due to a defect in glucokinase. Essential fructosuria is a disorder of fructose metabolism that results in hexokinase functioning as a main catalyst in metabolizing fructose through glycolysis. Alright, that wraps up hexokinase and glucokinase. Thanks for watching. If you enjoyed the video, don't forget to give a thumbs up.
Alright, now let's talk about glycolysis regulation and the key enzymes involved. Shown here, we see the glycolysis substrates, intermediates, as well as end products. I'll focus on the important steps, but I first wanted to offer a global picture of glycolysis. Notice how most of these steps have double-headed arrows, meaning that they can occur in either the forward or backward direction. Now what we're going to focus on are the unidirectional arrows, which we can see here. Here we can see the 3 steps of glycolysis that are irreversible, meaning that they only occur in the forward direction. And the net reaction of glycolysis can be shown in this equation. You start with a glucose molecule, you add 2 inorganic phosphates, as well as 2 ADP, as well as 2 NADH plus electron acceptors, and you end up with 2 molecules of each of the following: pyruvate, ATP, NADH, and a hydrogen ion. So what's important here? Well, you use 1 glucose to make 2 pyruvate, 2 ATP, and in this process, 2 NAD plus molecules get reduced to NADH plus a hydrogen ion. Now, if you get confused which species gets reduced or oxidized, don't freak out. Just look at the charge. Here, the NAD plus changes to NADH. So NAD plus loses its positive charge. In other words, its charge is reduced from +1 to 0 or neutral. Or to say this another way, NAD plus is reduced to NADH. Alright, now let's go through the enzymatic steps that are involved to regulate glycolysis. Now the 2 steps in glycolysis that require ATP are the first step and the third step, and you can see these here. This will represent our energy investment phase. First off, hexokinase or glucokinase will use 1 ATP to convert glucose to glucose 6-phosphate. And as we mentioned before, glucose 6-phosphate exerts negative feedback on hexokinase, but not glucokinase. Instead, high levels of fructose 6-phosphate act to inhibit glucokinase. Now, in the third step of glycolysis, phosphofructokinase 1 uses 1 ATP to convert fructose 6-phosphate to fructose 1,6-bisphosphate. And this just means that it's a fructose with a phosphate molecule at the 1 and 6 positions. And while we're on it, do you remember the rate-limiting step of glycolysis? There you go, it's phosphofructokinase 1, which we just talked about, which is the rate-limiting step of glycolysis. Now, since it's the rate-limiting step, there are going to be a lot of things that either activate or inhibit it. Some of these things include adenosine monophosphate, or AMP, and this just means that there's a lot of AMP around. This needs to be converted into ATP, so it's going to activate phosphofructokinase 1. Also, if there's a lot of fructose 2,6-bisphosphate, this indicates that the blood sugar is high, so there's plenty of glucose to be used for glycolysis, both of which are activators of phosphofructokinase 1. Now, if a lot of ATP and citrate are present, this means the cell has a sufficient amount of energy available and does not need to continue in glycolysis. Therefore, ATP and citrate will inhibit phosphofructokinase 1. Now, there are also 2 steps of glycolysis which also produce ATP. Both of these occur twice since they occur after the step where our 6-carbon molecule is split into 2 3-carbon molecules. This means that a total of 2 ATP are initially consumed and a total of 4 ATP are produced per molecule of glucose, resulting in a net gain of 2 ATP. Now that we've already invested some ATP prepping the glucose molecule, at which step do we actually make some ATP? Well, the first step in which glycolysis produces ATP uses the enzyme phosphoglycerate kinase, as you see here. Now, phosphoglycerate kinase converts 1,3-bisphosphoglycerate into 3-phosphoglycerate, and you can see this here. Now, going by the names of the molecules, it makes sense that 1 ATP is produced by this reaction, since 1,3-bisphosphoglycerate, which has 2 phosphates, is being converted into 3-phosphoglycerate, which has 1 phosphate. And remember that this reaction takes place twice, because halfway through glycolysis, glucose is split into 2 3-carbon molecules. And these 2 smaller fragments are called glyceraldehyde 3-phosphate, and they both undergo the same reactions to form 2 molecules of pyruvate. Now, the next enzyme that we need to discuss is pyruvate kinase. And pyruvate kinase is involved in the conversion of phosphoenolpyruvate to pyruvate, and this process also yields an ATP. And once again, as we just stated, this reaction will also occur twice for each glucose molecule fed through glycolysis. Now, what about the activators and inhibitors of pyruvate kinase? Can you think of these? There you go. Fructose 1,6-bisphosphate activates pyruvate kinase, and you can see this here. And if you think about it, this makes sense. If you have plenty of upstream substrate or building blocks for pyruvate, why not activate the enzyme that's going to make it? And on the opposite side of the coin, we see that ATP as well as alanine inhibit pyruvate kinase. And it also makes sense that if you have lots of ATP around, you would not have any reason to make additional ATP just for the sake of making it. But what about alanine? Can you think of why this might inhibit pyruvate kinase? Alanine is converted to pyruvate, and pyruvate can be converted into alanine using the Cow-Hill cycle of ammonia transport by alanine. And if you need a quick refresher, just go check out the Transport of Ammonia by Alanine video. So just like having plenty of ATP available, if you have plenty of a downstream product available, then there's no need to proceed down the pathway just to make excess for the sake of excess. Well, alright, let's take a shot at a quick flash quiz. In glycolysis, which reactions require ATP? Well, there are two steps in glycolysis that require the investment of ATP, and this is converting glucose to glucose 6-phosphate, as you see here, as well as converting fructose 6-phosphate to fructose 1,6-bisphosphate, as you see here. And if you think about it, it's just like investing on Wall Street. You need to initially invest some money, or ATP in this case, before you're ever going to get a return on your investment. And remember, that's just the 4 ATP that you get at the end of glycolysis. And just as a quick reminder, what's the net gain of ATP during glycolysis? There you go, there's a net gain of 2 ATP. Well, alright, that's going to wrap up glycolysis regulation. See you in the next video.